Hello everyone and welcome back to this online NPTEL course on aquatic geochemistry. In our previous lecture we have been discussing the application of different isotope systems as tracers in aquatic systems. We have started discussing the carbon isotopes. In this particular lecture we are again going to discuss carbon isotopes and along with nitrogen isotopes. So, in the previous lecture we were discussing how the carbon isotopes vary in the different systems, like in the carbonates that is getting precipitated in organic shells, to inorganic carbonates and also to that of organic matter, and how that helps us understanding the carbon cycle and the local productivity. And if I show some values, maybe that will make this point more clearer, where you can see here in this particular figure we have the delta 13 C values with respect to, of course, measured with respect to PdB, so of carbonates, and you see the natural variability for different for different N members, such as this is carbon monoxide, this is carbon dioxide. So, carbon dioxide in the atmosphere has this kind of variability. Okay. Then if you look into fossil fuels, you will see that they are slightly negative. Why? Because they are made up of organic matter, right? They are made up of dead decaying organic matter. So, they are very depleted in 13 carbon. Same thing for peat. Lake sediments have a wide range of variability. Why? Because there can be regions in the lake where there is a lot of anaerobic activities going on. So, they have dead decaying flux of organic matter. So, therefore, they are negative. Also, they are sometimes mixed with the atmospheric carbon dioxide. So, therefore, they have a huge range of natural variability in lake sediments. Then these are the different plant types, that is the C4 plant type and the C3 plant type. Okay. So, these are plants whose photosynthetic processes use different processes. So, there is a pathway called a C3 pathway and there is a pathway called a C4 pathway for photosynthesis, and according to those metabolic pathways, these plants are named either C3 plants and C4 plants. Okay. So, this is very important because it helps, as you can see, the C3 plants and the C4 plants have significantly different isotope composition. So, it is extremely useful. Okay. Methane again from decaying of organic matter will be negative. Okay, that is organic carbon, as compared, you compare here with hydrothermal carbon dioxide. So, hydrothermal carbon dioxide, that is coming from the mantle or from the mantle interactions with the ocean water. So, these are more fluidic interactions, and this carbon dioxide which is trapped inside the fluid inclusions. Okay, so, the water that got trapped around the mineral that got formed. Okay. This will have more or less the mantle kind of signature. Okay. So, this is enriched, or this is have enriched 13 carbon, because the 13 carbon that is there in the rocks are more stable, they are more in the the 13 carbon is preferred in those heavier stable forms. So, therefore, they are more of the positive values as compared to the organic ones. The graphite has a huge variation, diamonds have a huge variations, then there is carbon dioxide from volcanic gas. Okay. So, they basically show how the internal Earth's signature. Okay. So, similarly, there is morb, morb glasses, because glasses it crystallizes very quickly. So, a lot of fluid inclusions gets trapped, and these inclusions are taken out, they are sampled, and then their composition is determined. So, these give us very very like glances into what is happening deep inside the mantle processes. So, carbon in other igneous rocks, okay, and this is from Conrad. So, it is like the universe. Okay. Now, an extremely important understanding that we try to do is when we are trying to figure out how organic matter is how organic matter is getting transported in aquatic systems, and therefore we not only in aquatic system means terrestrial aquatic systems, also how it is getting modified in the coastal areas, how it is getting transported into the ocean and the deep ocean. Okay. So, for that we are often going to use carbon, and not only carbon, we are going to use it with nitrogen, because nitrogen, as I said, these group of elements, carbon, nitrogen, oxygen, these are the group that we are going to use when we are trying to determine influence of biomass and biotic interactions with the aquatic system. Okay. So, if we are trying to find out that okay, what is my terrestrial contribution, okay, and by meaning of terrestrial, if I also want to differentiate that which is my C3 plants, how much they are contributing, how much is my C4 plants they are contributing, if that kind of determination I can do, or if I am, so basically I want to separate out the different groups. I am using isotopes because they help me in separating, okay. They help me in looking into exact contribution from different sources. So, in that regard, this is extremely important and very, very useful. So, as you can see that this is the region or the data for C3 plants. So, C3 plants has this kind of variability in delta 13C isotope and in C/N ratio, and it is significantly the delta 13C value very nicely separates the two types in the range. Okay. Same thing for the different algae types also, whether if you are trying to look at a particular rock and you are thinking whether it was formed under lacustrine environment, that means lake environment, or whether it was formed under a marine environment, you are trying to redetermine this kind of paleo environment in which it was formed, then you may use delta 13C value of the carbonates, and you can see how if there is any biomass left or anything, and you can use those to determine whether you can find any marine algae or if you find any lake algae, because they are very have different values and very helpful in determining the kind of environment that it was. And certain if you have certain pitfalls, like whether there is any diagenetic impact or things like that, then these kind of ratios becomes very useful, okay, because these ratios do not get altered during some diagenetic effect. So, their ratios are always constant. Diagenesis may affect a particular element, but not the elemental ratio. Okay. So, therefore, if you are trying to if you are doing sediment studies, okay, you are trying to see what kind of environment that these sediments were deposited in, what kind of organic masses were supplying to the carbon here in this particular environment. So, this kind of isotope data becomes very, very useful. And in our aquatic systems, we are always trying to understand how much weathering is trying to the drawdown of carbon dioxide in a particular drainage basin, in a particular watershed, how much carbon dioxide is being drawn down from weathering, how much of it is getting transported from this basin to that basin, it is getting stored in the basin, what is the sources of this carbon, what is basically how carbon is getting transported through this aquatic system. So, that is our focus. Okay. So, when we are doing this kind of study, we are going to use the delta 13C of DIC values, okay, to understand the source contributions, and it is sometimes helpful to understand sources of the water itself. For example, if you have groundwater, if you have rainwater that is draining the soils, they will have very different kind of delta 13C values and DIC pools. Okay. And the different kinds of interactions, for instance, if there is respiration going on, what will happen? The DIC will increase, but the delta 13C values are getting lowered. Why? Because with more respiration, my 12 carbon is not coming in, my 13 carbon is not coming, my 12 carbon is coming as gas, right. So, my delta 13C is getting lowered, but since more and more carbon dioxide gas is getting incorporated and dissolved, then it will form DIC, right. So, it will, so CO2 plus H2O will form HCO3, it is going to form that bicarbonate acid. So, this is the reason why during respiration we see that the DIC value is going on increasing and the delta 13C value is going on decreasing. Same thing for carbon dioxide invasion also, that if carbon dioxide is getting if it is taken from the atmosphere, if it is getting soluble taken down, drawing down from the atmosphere, the delta 13C value is getting lowered and the DIC is getting increased. And just the opposite is happening during photosynthesis. Why? Because now my carbon dioxide is getting incorporated in the biomass. So, my carbon is going into the biomass. So, my DIC values are getting lowered, but my delta 13C values are getting increased. Why? Because 12 carbon is preferentially incorporated in the biomass. So, during photosynthesis, 12 carbon preferred in the biomass. So, whenever there is a huge photosynthetic event happening, or there is a huge algae bloom is happening, there will be rapid drawdown of DIC values, while the delta 13C values are increasing. So, such changes are happening. For instance, if you are looking at groundwater, okay, groundwater is interacting with rocks, the carbonates in this system, the carbonates in that aquifer, rocks in that aquifer region. So, the delta 13C values of the DIC is going to be higher, because rocks generally have more 13 carbon. This 13 carbon is preferred in the stable phases. So, the DIC is also high, the 13C carbon value is also high. So, that is how we try to understand, okay, if there is a contribution coming from groundwater suddenly, okay, if the how is the carbon dioxide cycle happening and the DIC pool is changing in the river itself. So, when we are looking into plots like this, okay, where we have the delta 13C value measured across a year. So, there are seasonal changes we can see. So, during the spring there is some formation, the values are getting higher, then again get lower, again it is getting more low, further low during winter. So, what is happening? Why is it getting changed? Why is the delta 13C value also going to change? Why is the DIC value is also going to change? So, we are continuously trying to use these data sets to understand the contributions of different tributaries, of different water sources such as groundwater and surface water mixing. If there is local soil pools, then how much they are contributing. So, such kind of information we are trying to derive using such kind of data sets, and therefore, delta 13C of DIC measurements is extremely common for aquatic studies and of immense applications. Helps us identify a lot of different kinds of processes that is happening in the basin. Now, let us look into the nitrogen isotopes, how nitrogen isotopes are helpful. So, nitrogen is a trace phase in the rocks, okay. It is a trace phase in the rocks, it is not a lot in the rocks, but and a major component of the air. Of course, as you all know, it is a major component of the air, but still if you consider all the nitrogen on the planet, okay, if you consider all the nitrogen on the planet, 97.9% of all the nitrogen is there in the rocks. That is there in the rocks, that is trapped in the rocks. Okay. The pool that we are living in, that is the air that we are circulated around, okay, is only 2%. Okay. The nitrogen pool that is in the air, that is makes only 2%. Out of the 2%, only 0.01% is in organic matter, that is we are made up of. Of that 0.001%, again 60% is dissolved in the ocean, that means organic matter that is dissolved in the ocean, and from that 0.001%, 25% is there across the globe as soil organic matter, and only 1.5% is there as living biomass. So, just imagine the small fraction of nitrogen that is there in living biomass. Okay. Now, when we measure nitrogen, we are measuring nitrogen with respect to air. Okay. So, delta 15N means 15N by 14N of the sample divided by 15N by 14N of the air minus 1 into 1000. Okay. And here you can see that the nitrogen delta 15N value in sedimentary rocks is around plus 10 parts per mil, while air we have the value of 0. So, just like for water, we have VSMO, the ocean water, and ocean water has a value of 0. For air, we have a value of 0 parts per mil of delta 15N. In this right hand side figure, there is a plot showing the difference of delta 15N values and the different kinds of reservoirs in earth. So, here we have atmospheric nitrogen and its value that we have, that is 0 parts per mil. Then there is ammonium, that is oxidized form, or as nitrate reduced form, and how its natural variability are. As compared to that, look at the natural variability in the deep ocean nitrate, it is very narrow range of variability. Similarly, oceanic organic matter, POM is particulate organic matter. That means it is not in the dissolved phase, it is there as solids, as particulates. Then this is the variability in plants that we observe. This is the variability we observe in animals, and with increasing trophic level, we are going to move along the more positive side. Soil organic matter again has huge variability, and then we have soil ammonium and soil nitrates. These are fertilizers, inorganic ones and organic ones being more positive. Then again a huge variation in the natural gas that we have producing from organic matter decay. Petroleum reservoir that has this kind of 15N value, volcanic gases has this kind of value, morb has this kind of signature. This is that of felsic continental rocks. Again diamond has a huge variability, while meteorites can have this variability or moving towards more and more positive side, that means extremely stable form, very high δ¹⁵N values. But question is, what is controlling these variabilities and why do we see such variabilities? Now, although the organic nitrogen is minor, that means as compared to the rock nitrogen, it is a very minor in abundance, only 1.5% of the 0.001%. Now, this small contribution also is very important, because all nitrogen isotope fractionation occurs by metabolic or metabolically related processes. Okay. It is due to metabolic process, meaning all the fractionation, all the differentiation that is happening in the isotopes is mostly related due to the metabolic activities that it is undergoing, that the organic matter is undergoing, and very rarely due to other other parameters such as temperature or interactions with other inorganic reactions. So, this is mostly metabolic and metabolically related processes. For instance, what are the processes we are talking about? We are talking about nitrogen fixation, that is when nitrogen is removed from the air by microorganisms and it is made available to the plants, that is the nitrogen fixation process. Then there is assimilation, when that fixed nitrogen either in the ammonium form or the nitrate form gets incorporated in the tissue, it gets uptaken into the biomass, then this process is called assimilation or immobilization. Now, the nitrogen is immobilized. The reverse of this process is called degradation of organic matter, which is done by the heterotrophic bacteria, and the ammonium is again released back into the ecosystem. So, the process is called either mineralization or ammonification. Then there is a process called nitrification, where ammonia can also get oxidized, okay, the oxidation of ammonia to nitrate. Then there is another process called denitrification, where the nitrate gets reduced to nitrite, and they can further get reduced to various kind of nitrogen oxides and ultimately to nitrogen gas by the anaerobic bacteria, that is some kind of fungi and other bacteria, and this nitrogen is released back into the atmosphere. So, these are all the degradation of organic matter, okay, that is from ammonia to nitrate, from nitrate to nitrite, from nitrite to nitrogen dioxides, and ultimately to nitrogen gas, and then release it back into the atmosphere. So, these are the entire processes. But when we look into variations, what do we see? That these processes cause isotope fractionation that are difficult to quantify. When we were looking into 13 carbon, the diagenetic effect, the vital effect meaning the δ13C values that is changing due to the metabolic metabolic activities, they were acting only for those animals, who who were using, whose impact of metabolic activities was significant on its carbonate shell formation. And since we have an entire separate group of inorganic carbonates, metabolic activities becomes irrelevant in that form. So, we have a very strong proxy, the inorganic carbonate, that does not suffer from any kind of metabolic activities. Well, even though a small fraction of the carbonate isotopes of the carbonate samples, they can be they can have fractionation due to this vital effect, but even then this fractionation is very constant. Like even though there is some changes due to some metabolic activities, it is very stable, like we can do some corrections to change the δ13C values. But for nitrogen isotopes, this is very difficult to say, because of the multiple pathways, because of the multiple forms that it exists, that is from ammonia to nitrate to nitrite to the different nitrous oxides. And since it is so, it is a fundamental block for organic matter, the different processes that it is undergoing, the transformations at metabolic activities at each stage is difficult to identify, quantify, and these are all driven by kinetic processes. They are not in any equilibrium. There is no equilibrium concept when you are in a metabolic process. That means, if you have good nitrogen nitrate supply, you are going to do this, do that. If the nitrate supply drops in a single minute, your process is going to change. And the fractionations vary at each step. It is so variable. It depends plus on the ambient conditions. That means, where is the nutrient coming from, what kind of bacteria is there. So, for each setting the data is different, such that it is unable, we are unable to have a very particular values for particular processes, because as you can see, the frequency in kinds of assimilation of different groups of nitrogen compounds and their entire range. So, they are like all over from -16 to 0 and to 4, and within the 0 to -2 we can have ammonium also, we can have nitrate also, we can have nitrite also. So, it is because of this kind of variability we may not do very specific, source specific and process specific studies. Okay. And also the huge kind of processes. So, for example, this kind of variability is arising because of what? Availability of the nitrogen itself. For instance, if you are in ocean, nitrogen is one of the limiting nutrients. If it is there, it becomes one of the limiting nutrients. That means, productivity gets limited due to the lower supply of nitrogen. So, it is called one of the limiting nutrients. Then there is various enzymes that is responsible for NH3 fixation. These enzymes can vary across organisms. It is very kinetic. So, how it will show up you will not know. Plus all the kinetic diffusion that is happening through the cell walls, different organisms, different ambient conditions. Due to such variability, there is a wide range of values that we observe, wide range of values we observe. Let us say the nitrogen cycle itself. So, we have nitrogen gas that gets fixed. So, there is a labile organic nitrogen. From the labile organic, a certain proportion gets into the stable form. It is there in the stable biomass, while the labile organic is there in the soil. Okay. It's ready for uptake, but not yet uptaken. Okay. That through different bacteria and all that can again get converted into ammonium. Through ammonification can get converted to ammonium. Ammonium gets converted to nitrite, or it can get converted to nitrate. Nitrate will definitely convert back to nitrite, and then through denitrification process get released as nitrogen gas. In all the brackets you see the values that is there from one source to one sink. So, if this is the delta 15N of ammonium, this is the delta 15N of nitrite value. Okay. So, therefore, you see the huge range of values that can happen. The different processes can have leads to different kinds of values. Okay. Now, denitrification tends to occur in deeper layers of soil or in poorly aerated soils. Why? Because there we have more anaerobic bacteria. These anaerobic bacteria will use this denitrification step to gain oxygen, and then to get the energy for its metabolic activities. Similarly, in the ocean, denitrification is active in stagnant water, or where the dissolved oxygen itself is so low that the bacteria has to depend on anaerobic processes such as denitrification processes to get the energy. So, denitrification increases with depth in the ocean, okay, and without nitrogen getting supplied back into the atmosphere, we would not have had any nitrogen left itself. Okay, it would have been got exhausted in the first 100 million years. It is just due to this continuous denitrification processes, continuous release by the ocean mass, the ocean organic matter that is getting released back into the ocean from the terrestrial ecosystem, marine ecosystems, that we still have nitrogen as a major gas in the atmosphere. Okay. And how is it varying? How is it varying? Nitrogen, as I have already told you, again prefers the 14N is preferred as compared to the 15N. Okay. So, with depth, you see the denitrification process is highest here. Okay. So, nitrates are more, as it is moving up, it is getting denitrified and it is moving up. So, nitrate composition in the water column, and correspondingly its delta 15N values. So, in shallow aquifers, nitrogen gas produced by the denitrification is lost by diffusion to the atmosphere, and this is a continuous process just like the classic Rayleigh fractionation process. Similarly, as you can see here in the ocean, this is the data from the North Pacific Ocean, when nitrate is getting converted to nitrogen, and as it is happening, its delta 15N value is increasing. Okay. And due to different kind of fractionation factors, we sometimes do not know what corrections we can make. So, this is showing a trial and error method, where you give that there is a 30%, 40% difference between the 15 nitrogen of the nitrate and the nitrogen gas that is being produced, and you see which line fits your data. The delta 15N values for the animals is here. So, for the marine plants and animals is a function of the trophic level, and at each trophic shift, you can see there is an increase of 3 parts per mil per trophic level. Why? Because it is getting more and more complex, more and more stable. So, the delta 15N values are going to get more and more enriched. Delta 15N values are getting more and more enriched as the trophic levels are increasing. So, by combining different kinds of data sets, now when we are looking into aquatic systems, okay, when we are looking into aquatic systems, so for instance, this is the nitrogen system for the oceans, and we are looking into particulate organic matter, how it is interacting in the water body, all this when we are looking into, then this becomes a supporting evidence when we are using it with some other kind of isotopes such as carbon or such as oxygen, because we know that if there is oxygen is there, and we can expect some CO2 is also evolving in that data. So, not only we are going to measure nitrate values and delta 15N values, we are also going to measure DIC values, and we are going to measure the delta 13C of the DIC values. So, the sources and the sinks in oceans, how it is evolving, okay, it is important to use combined data sets, okay, and also with the different plants and animals, how it is interacting. So, this is a change of the trophic level, this is the proportion of C4 in the diet. So, these are the different animal groups, these are the different groups of humans, actually it is Native American communities. So, how delta 15N values and delta 13C values together can help us identify what was the diet of that particular community. When we are doing paleo studies, when we are doing archaeology, okay, these kind of data sets becomes extremely important. What kind of diet were these people having when they were migrating? What kind of migration was it? How the different populations were mixing. So, these combined data sets is extremely useful, and since most of the time it gets mediated through aquatic systems, the marine system, lakes, rivers, understanding their behavior is very, very important. So, with this I come to the end of this lecture today, and in our next lecture we are going to see some more isotope systems and how we apply them in understanding different kinds of processes and sources and these things. Thank you.